The central dogma of molecular biology states that DNA leads to RNA and then it leads to proteins. But what does that mean? Your cells, they carry chromosomes that are made of DNA, but the DNA only holds instructions for making a protein. That's all, it's just info storage. There is a process called protein synthesis in which the DNA is accessed for its instructions, like a recipe, and then the instructions are used to build a protein. So that explains the blue DNA on the picture on the right and the purple amino acid chain, but what's the pink? In this process, there's an intermediary that's used to carry the information from the nucleus in the DNA to the place in the cytoplasm where the protein will be assembled according to the instructions. All right, so I have Swedish lineage, and I have my great-great-grandmother's cookbook. It's in Swedish. I needed it translated, and doing it myself was a little slow going. So I found a woman on the internet that did translation services for Swedish to English. And unfortunately, she was on the West Coast, but I didn't really care where she was because I was not sending her my great-great-grandmother's cookbook. I made a copy, and I sent her the copy, and then she translated it, and then she sent me back the English version. And I then made wonderful Swedish recipes like curried egg salad and salted cod. Yeah, great. But the point is that I retained the original, like the DNA that stays in your nucleus, and I sent out a copy to be translated into this new language, just as RNA, or that pink on the diagram, carries the instructions for making the protein to be translated from nucleic acid to amino acids. What does this mean for you? Your DNA holds the instructions for the proteins that make you, you. For example, there are pigmentation genes in your chromosomes. They carry instructions for making a pigment of a certain color. And then that pigment can be made and inserted into your eyes to be expressed as your eye color for the world to see. Recall from the DNA replication mini lecture that we define a mutation as a permanent variation in DNA that happens because of damage to the DNA. Again, yes, these can result in cancer, especially if they target the right genes in the cell. There are different kinds of mutations, and I have you consider the effect of these different types in the DNA transcription and translation lab. In that lab, we consider the effect of each type of mutation on the protein for which a gene codes. We previously described these general types of mutations. First, we had a point mutation, where one nucleotide is substituted for another. Second, we had frame shift mutations, where we inserted or deleted a nucleotide. Without even describing these, I'm sure that you can see that mutations change the original DNA sequence, which changes the codon, the anticodon, and possibly the protein. No matter what type of mutation occurs, substitution or deletion, if the mutation doesn't cause any difference in the resulting protein, it is called a silent mutation. How can this happen? Well, take a look at the genetic code. In general, if we change the last nucleotide on a codon, it probably doesn't change the amino acid. Do you see that changing CUU to CUC doesn't change the fact that leucine will be the amino acid in the protein chain? You are accumulating silent mutations right now. You get more and more of them as you age. Let's do a missense mutation. Just think of it like using wheat flour to make bread instead of using white flour. You still get the bread, but it's not really what you intended. Every human has a gene that says, make a red blood cell. Actually, it's actually a bunch of genes that say make a red blood cell. But to keep things simple, let's just say, for example, that it's one gene that determines if your red blood cells are nice and plump, like the one on the top of the picture here, and thus they're able to carry oxygen, or if your red blood cells are sickle-shaped, like the C-shaped red blood cell below it, and that red blood cell is not so great at carrying oxygen. There is a variant of this gene in the human population where one nucleotide has been changed. One nucleotide. Somewhere in the sequence of DNA, a thymine got replaced by an adenine. Oops. But the changes the codon, which changes the anticodon, which changes the amino acid to which the codon matches. In people with sickle cell anemia, there is one amino acid out of thousands. That is the genesis of their disease. But what if the nucleotide changed and it didn't lead to a different amino acid? Great, fine. That's what's called a silent mutation. Let's do some mutations here with the Maggie protein. This lab, the lab associated with using the genetic code, it gives you a sequence of DNA that will make this Maggie protein, and that has only 4 amino acids in it. So it's nice and simple for a lab. Let's figure out what those 4 amino acids are by transcribing and translating this DNA sequence. We can use those base pairing rules to figure out the RNA sequence that starts with AUG, that all amino acids start with, right? Don't forget that when you transcribe, you can't use T in the RNA, right? Don't forget the base pairing rules. Let's translate the first 2 amino acids using the genetic code. Don't try to read this little tiny genetic code that I have here on the slide. Try to, print out your own. To start, let me give you a hint. Methionine, MET, is the first amino acid in every single protein that you will transcribe. Please don't forget that. I mean, it kind of is the first amino acid because sometimes after the protein is made, methionine can get cut out upon final folding of the protein. But look at your genetic code. AUG matches with the methionine amino acid. Keep going. Find GCC on this genetic code on the right. What is the corresponding amino acid? Keep doing this until you get to the UAA codon. That corresponds to a signal to stop making the protein. So what is the GCA at the end? Well, it's kind of junk. I mean, kind of. Let's just say that addition of DNA there, it's explained in a future mini lecture. Just consider it non-useful right now, which is fine. Let's apply the Maggie protein to the different kinds of mutations. In the lab, you are given these variations of the Maggie protein DNA sequence. The first one listed is the one that we just translated into met, ala, gly, and tir amino acids. Look at the silent mutation, where that one amino acid is circled in red is substituted for the original A. Translate this DNA sequence using the process lightly described in the previous slide and see what happens to the protein. Remember what a silent mutation means, that there will be no difference in the resulting protein. Hint, 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 hint. Check out the missense mutation that is also just one nucleotide, but this time it makes a big difference. And we have a frame shift mutation that causes complete rearrangement of the codons. This example here is when one nucleotide is inserted. But remember that frame shift mutations can also be deletions as well. For the lab, you have to translate these 4 DNA sequences. Remember to make the RNA first, and then use the genetic code to determine the amino acid.